Hello everyone. I welcome you to the session of Ask Me Anything from Biology. Okay, so let me introduce myself. I am Mamalin Mohanty. I'm a second year MBBS student at SCB Medical College and Hospital. I have cracked NEET in 2022 with All India rank of 2101, and now we'll start. Okay, so we have received immense response from you guys, and we have shortlisted 22 sort of questions which we'll be answering in this session. Okay, so if you have any doubt, then do comment. I will try to answer them at the end. Okay, so let's start. First question is, what is the different glucose breakdown pathway? Okay, so as we know, for all living organisms, glucose, it is the most significant source of energy, right? The carbohydrate we take or we eat, which the most essential of which is glucose. Okay, the glucose get oxidized. It is oxidized to give what? Energy. Okay, so it has two pathway. One is aerobic pathway, one is anaerobic pathway. Okay, aerobic pathway, as we know, it requires the presence of oxygen, and anaerobic pathway, it occurs in absence of oxygen. Okay, so in both aerobic and anaerobic pathway, the common pathway is glycolysis. Okay, it is present both in aerobic pathway and anaerobic pathway of glucose breakdown. Okay, so in glycolysis, what is happening? The glucose first turned into glucose, which is a 6-carbon compound. Okay, it get converted into 3-carbon compound, which is pyruvate. Okay, pyruvate, it is a 3-carbon compound. So one glucose molecule will give rise to 2 pyruvate molecule. One glucose molecule will give rise to 2 pyruvate molecule. Okay, it is the common pathway. So glycolysis, what are the steps? The first step is conversion of glucose to glucose 6-phosphate. Okay, I hope it is visible. Okay, conversion of glucose into glucose 6-phosphate. It is the first step in presence of hexokinase. Okay, second step is conversion of glucose 6-phosphate into fructose 6-phosphate in presence of isomerase. Okay, next is fructose 6-phosphate to fructose 1,6-bisphosphate in presence of phosphofructokinase. Okay, in the first step and the third step, ATP is being utilized. Okay, ATP is converted to ADP, right? In the fourth step, fructose 1,6-bisphosphate is converting into glyceraldehyde 3-phosphate and dihydroacetone phosphate, dihydroacetone phosphate, DHAP. Okay, it is a split reaction, right? Then the glyceraldehyde 3-phosphate is converting into 1,3-bisphosphoglycerate. Okay, here there is a generation of NADH2 molecule, right? Then the next step, 1,3-bisphosphoglycerate is converting to 3-phosphoglycerate. Next step is 3-phosphoglycerate to 2-phosphoglycerate and 2-phosphoglycerate to phosphoenol pyruvate. Okay, then the last step is phosphoenol pyruvate is converting into pyruvate molecule, right? It is the common pathway between aerobic and anaerobic respiration. So in aerobic respiration, okay, first is aerobic respiration. In aerobic respiration, what is happening? The first step is glycolysis, right? First step is glycolysis. Second step is Krebs cycle. Krebs cycle. Then the third step is electron transport chain. Okay. Third step is electron transport chain, ETC, which is the oxidative phosphorylation reaction. Okay. It is oxidative phosphorylation. That means here NADH2 and FADH2 molecules they will generate ATP. Okay. Then, okay. So first step, glucose is converted into pyruvate during glycolysis. Then the pyruvate produce carbon dioxide and water along with energy. It is a aerobic respiration. Okay. Then in anaerobic respiration, here let me write this. Anaerobic respiration. Okay. The process takes place in absence of oxygen. Right. Then there are two, this is a type of fermentation reaction. Okay. Two types of fermentation can occur in the absence of oxygen. First one is alcoholic fermentation. Okay. Alcoholic fermentation. Second one is lactic acid fermentation. Lactic acid fermentation. Okay. In alcoholic fermentation, okay, alcoholic fermentation occurs in yeast. It occurs in yeast. In absence of oxygen, as we all know, it is an anaerobic process, right? Here only glycolysis occurs, and at the end of glycolysis, the pyruvate is produced from the glucose because glycolysis is the common pathway, right? Then the pyruvate is produced in the process of glycolysis. Then the pyruvate is converted to, here, the pyruvate. It is converted to what? Ethyl alcohol. Okay. This is the process of alcoholic fermentation. Okay. Then what is happening in lactic acid fermentation? Okay. It occurs in the system due to lack of oxygen, right? Then it occurs in the muscle. Alcoholic fermentation occurs in yeast, whereas the lactic acid fermentation occurs in muscles. Okay. When we do vigorous exercise, then our muscles do not get enough oxygen. Then anaerobic respiration takes place in our muscles and lactic acid fermentation takes place. Due to lactic acid, we get muscles cramp. Okay. Due to lactic acid, we get what? Muscle cramp. Okay. Now see this picture. Glucose, it is converted to pyruvate molecule in the process of glycolysis, right? Then in absence of oxygen, that is anaerobic process, it what? 2 ethanol or lactate molecule is producing. Okay. Then 2 pyruvate. The pyruvate molecule in presence of oxygen, that is aerobic, in presence of oxygen, that is in aerobic respiration, it is converting to 2 acetyl CoA. Then it goes to Krebs cycle. Formation of 4 carbon dioxide molecule takes place. Then the NADH and FADH2 molecule, their oxidative phosphorylation takes place and ATP is being produced from NADH2 and FADH2, right? So now we'll solve the question. What is it? What it is written? The glucose, that is 6 carbon molecule. It is converting to pyruvate, that is 3 carbon molecule, right? The process is known as glycolysis. It takes place in the cytoplasm. In the cytoplasm. Okay. Then the pyruvate 3 carbon molecule, it is converting to 2 carbon molecule, carbon dioxide and energy. What is this 2 carbon molecule? We know that this ethyl alcohol, it is a 2 carbon molecule, C2H5OH, right? So this is the ethyl alcohol or known as ethanol. Okay. It occurs in absence of oxygen and it occurs in yeast. Okay. Then lack of oxygen in our muscles. Muscle cell, what is happening? Lactic acid fermentation happens in our muscle cell in lack of oxygen. Okay. What are the byproducts? The byproducts are energy plus lactic acid. Okay. So now aerobic respiration, that is in presence of oxygen, it is taking place in the mitochondria. What are the byproducts? One is energy, one is carbon dioxide, and one is water. Okay. So now we know the different glucose breakdown pathway. Okay. Is it clear? Then we have our next question. What are gastric juices? Okay. As we all know, gastric juices produce in our stomach. Okay. It is our stomach. Then it is our esophagus. Saliva produced in our buccal cavity, right? In stomach, gastric juices is produced. Then and digestion takes place. Okay. What constitutes the gastric juice? First is about 1 to 1.5 to 2 liters of gastric juice produced every day in our stomach, and it consists of, first one is water, okay, second one is mineral salt, then third one is mucus, which is secreted by mucous neck cell in the gland and the surface mucous cell in stomach surface. Okay. Then the fourth and important one is hydrochloric acid and intrinsic factor. They are secreted by the parietal cell. Okay. Last one is the inactive enzyme precursor. These are the pepsinogens which are secreted by the chief cell in the gland. Okay. So we'll see the function of gastric juice here. First, the water. Water further liquefies the food we swallow. Okay. Then next function, the next important function is the hydrochloric acid. Okay. First, hydrochloric acid, it provides a acidic medium. Here you can see the stomach acid, that is HCl. The pH is ranges from 1 to 2, which is strongly acidic in nature. Okay. So the hydrochloric acid, it provides a pH of 1 to 2, that is acidic medium, where the pepsinogen, here the inactive enzyme precursor, pepsinogen, that is converted to pepsinogen, is getting converted to pepsins in acidic pH. Okay. Right. Then pepsin, what it does? It breaks down the protein molecules. Okay. Where does the gastric acid is secreted from? It is secreted from our parietal cell. It is one important question. Okay. Next one is then gastric acid kya karta hai? Gastric acid plays a key role in the digestion of protein. Okay. Gastric acid, it is providing a acidic pH medium where the pepsinogen is converting into pepsin. Then pepsin has a key role in the digestion of protein by activating digestive enzyme. It is the activated digestive enzyme. Okay. ये क्या करता है? It breaks down the long chain of amino acid of protein. It breaks down the long chain of amino acid. Okay. So the first function of hydrochloric acid is it acidifies the food and stop the action of salivary amylase. First function. Second function, it kills the ingested microbes. Okay. Then third function is it provides the acidic environment that is needed for the action of pepsins. Okay. Then pepsinogen, it is converted to pepsin in acidic medium. Then it breaks down the long chain of amino acid of proteins. That is, it helps in protein digestion. Okay. Then next function is of intrinsic factor. Okay. Intrinsic factor क्या कहता है? It helps in transportation and absorption of vital micronutrient such as vitamin B12. Okay. What it does? It helps in transportation and absorption of vitamin B12. Last function is mucus. Okay. It is a protective mechanism by our stomach. Okay. It provide protection from the acidic medium. Right. Then how does the gastric acid or gastric juice is secreted? Okay. There is always a small quantity of gastric juice present in our stomach, even when it contains no food. This is known as fasting juice. Okay. Secretion reaches its maximum level about 1 hour after the meal. Okay. When our digestion takes place, right? There are 3 phases. One is cephalic phase. That means when food will be in our mouth, then the amount of gastric acid secreted, it is known as the cephalic phase. Then gastric phase. Then when food is inside our stomach. Then intestinal phase is when food is inside our intestine. Then the amount of gastric acid secreted is known as, the phase is known as intestinal phase. Okay. Saliva, that is in buccal cavity, it is cephalic phase. In stomach, it is gastric phase. Okay. And in intestine, it is intestinal phase. Intestinal phase. Okay. Clear? Then we'll go to the next question. It is, what is holozoic nutrition? Okay. If we'll break down this word, then first one is holo. Holo means whole. Okay. Then zoic means it is of animals. That means it is a holozoic nutrition, means it is a heterotrophic nutrition. Heterotrophic nutrition. Okay. That means they do not prepare their own food. Okay. They depends on the other animal for their food. They are heterotrophic animal and the nutrition is known as heterotrophic nutrition. Right. It is characterized by the internalization, that is the ingestion of food, and internal processing of liquid and solid particles. Okay. What are the examples? Okay. First is protozoa. Okay. Protozoa, the example is here, amoeba. Right. You can see the parts. This is the pseudopods. This one, it is the contractile vacuole. Contractile vacuole. Then this is the endoplasm. Next, this is nucleus. And this is the cell membrane. Okay. Protozoa, that is amoeba, it shows what? Holozoic nutrition. Okay. Then next one is one free living organism, that is us, human. It shows human shows what? Holozoic nutrition. What are the steps that involves in holozoic nutrition? First one is ingestion. Okay. Ingestion means the food is first taken into the body through an opening known as mouth. Okay. Second step is digestion. Then the food is converted to simpler form or soluble form by various enzymes in different part of our alimentary canal. Okay. The process is known as digestion. Next, the third process is absorption. Absorption means the simplified product that formed, they are being absorbed. Okay. Fourth step is assimilation. That means the conversion of nutrient into fluid or solid substance of the body. Okay. And the last step is egestion, that is excretion, where the undigested part of the food is removed from our body. Okay. We have 5 steps in holozoic nutrition. Right. Next step is, next question we have, write the name of the solution used to preserve biological specimen. Okay. You all have seen this type of specimen preservation in your science lab. Okay. The specimens are being preserved in a chemical. The chemical is known as what? Formalin. The chemical is formalin. That is 40% formaldehyde by volume and 37% formaldehyde by gas. You only have to remember the name, that is formalin, which is a formaldehyde, that is HCHO. Formaldehyde. Okay. What is the function of formaldehyde then? First function, it is a strong disinfectant. Okay. Next is, it is a tissue hardener. Okay. It is tissue hardener. Then third one is, it is a germicide. Okay. And fourth one is, it is antiseptic in nature. It is antiseptic in nature. Due to this characteristic, formalin is used to preserve specimen in all the labs. Okay. So then we will proceed to our next question, which is, what are auxins and what are gibberellin? Okay. First, auxin and gibberellins, they are the PGR. What is PGR? It is plant growth regulator. Okay. Okay. Auxin, it occurs naturally as well as artificially. Okay. It occurs naturally. It can be artificial as well. Okay. What is the main function? The important role is to regulate plant growth. It regulates plant growth. Okay. One important point is auxins first, that is initially associated from human urine. It is a neat question that auxin were initially isolated from human urine. Okay. First, we'll see the function of auxin. First point is, auxin are specific group of hormone that plays important role in plant growth and development. Okay. Second function is, it synthesized at root tip in the presence of sunlight. Okay. It is not the function, these are the characteristic feature. Third point is, the term auxin is derived from the Greek word auxein, means to grow or increase. Fourth is, it is the first plant hormone to be discovered. Okay. The most common auxin is IAA, that is indole acetic acid. This is the chemical formula which you need not to be known. Okay. Then we'll know the functions of auxin. Okay. I write first function. These are the main functions of auxin. First function is cell elongation. Okay. Auxin promotes cell elongation of shoots and coleoptiles. Okay. Cell elongation of shoots and coleoptiles. Second point, it helps in cell division and differentiation. Okay. It promotes healing. First point is it promotes healing and regeneration of vascular tissue. Then what are the vascular tissue in plants? It is xylem and phloem. Okay. Next point is callus formation. Okay. Auxin along with another PGR, which is cytokine, it induces callus formation. Okay. Fourth point is secondary growth. Okay. It induces cell division in vascular cambium, and vascular cambium, it is of secondary growth. Okay. It induces cell division in vascular cambium. Then we have the fifth function. It is root initiation on cutting. Root initiation. Okay. For asexual propagation, NAA, that is, it is the synthetic form. It is used to initiate root formation. Okay. Then sixth one is apical dominance. Apical dominance. Okay. So, this is a plant. Okay. If there is an axillary bud here, then it inhibits the length of apical meristem. Okay. This phenomenon is known as apical dominance. Okay. If the shoot tip it is removed, then it induces what? It induces formation of lateral buds. Okay. And it is used to promote what? Branching. Right. It is used in hedge making, and tea plantation. Okay. Here the shoot tip is removed, which induce the growth of lateral buds, and it also promote branching. Okay. Seventh point is parthenocarpy. Okay. When auxin is applied to some flowers, it induces what? Parthenocarpy. Okay. What do you mean by parthenocarpy? The ovary gets enlarged and they develop into seedless fruit. Parthenocarpy means formation of seedless fruit. Okay. Here also gibberellin helps it. Gibberellin stimulates the fruit development. Okay. So you can add another function, that is fruit development. Okay. Then ninth point is it delays senescence of flower. It delays. Okay. Then the last point is it acts as herbicide. The synthetic auxin. What are the synthetic auxin? It is 2,4-D and 2, 4, 5 T. Okay, these are widely used to kill weeds. Okay, one point you remember that it doesn't affect grasses. Okay, okay, it's all about auxin and its function. Then we'll come to gibberellin. First point is it promotes pollen development and pollen tube growth. Okay. Second point is it promotes fruit setting and parthenocarpy. Third point is it promotes seed germination. Then it enhances alpha amylase activity. Okay. It is not that important for you. Okay. Next point is it enhances the calcium, phosphate and potassium content. Then it regulates transition from juvenile to adult plant. Then last point is it stimulates stem and root growth. Okay. Some of the important NCERT points that I want to highlight for gibberellin. That is, gibberellin is a promotory PGR. We know. Okay. There are more than 100 gibberellin that is reported from widely different organisms, such as fungi or any higher plant. Okay. That denoted as GA1, GA2, GA3, and so on. GA means gibberellic acid. Okay. Then you say, which is the first gibberellic acid to be discovered? It is not the GA1, it is GA3. Okay. It is the first gibberellin to be discovered and it remains the most intensively studied form. Okay. It is the GA3. Right. So, you know, the full form is gibberellic acid. That means it is acidic in nature. Okay. What are the physiological response in plants for gibberellin? First, it increases the stalk, increases the length of the stalk, length of the stalk in grapes. Okay. These are the grapes. It is the stalk. It increases the length of the stalk. Okay. Next point is the fruits like apple. It elongate and improve the shape of fruits like apple. Second point, it elongate and improve the shape. Okay. Next point is GA3, that is gibberellic acid 3. It is the first gibberellic acid to be discovered. It is used to speed up the malting process in brewing industry. It is a potent question for NEET. What? Gibberellic acid, that is GA3. It is used to speed up the malting process. In which industry? In brewing industry. Okay. Then, you know, sugar cane. This is the sugar cane. These are the nodes. Okay. The sugar or the juice that is stored in this part, right? If we'll increase the length of this part, then the productivity or the production of the crop will be increased, right? The gibberellic acid, that is gibberellin, it increases the length of the stem in sugarcane. It increases the length of the sugarcane stem. Okay. Then last point is, okay, last point is it induced early seed production. What it induced? It induced early seed production. So these are some function of gibberellic acid, that is gibberellin, and these are the function that is written in NCERT. Okay. So next question we have, what is the scientific name of mango? Okay. First, the scientific name of mango is what? Mangifera indica. Okay. Now we'll see in Mangifera indica, it is the genus for Mangifera is genus, indica is species. Okay. The first alphabet of genus should be in capital letter. Okay. Whereas the first letter or first alphabet from species, it is written in small letter. Okay. And the scientific name is always written in italics, and it is separated. It is underlined separately. Right? How to write a scientific name? Again, I am telling the genus part, the first alphabet of genus part should be written in capital, and the first alphabet from the species part should be written in small letter. Okay. The whole scientific name should be written in italics and it should be underlined properly. Okay. So what we have? Mangifera indica. It is the scientific name of mango. Okay. Now, can you tell me what is the family of mango? It is always and it is mostly asked in NEET. Okay, in PYQ. So, what is the family? Mango belongs to which family? The answer is what? Anacardiaceae. Okay, it is also a potent question. Okay, so we have done question number 6. What is the scientific name of mango? Then the 7th question we have is define parturition and lactation. Then what is parturition? Okay, it is the action of giving birth to young. That is the childbirth. Okay, it is the action of giving birth to young or childbirth. It is the action of giving birth. Okay, so what is written? Parturition is a process by which the fetus, this is the fetus, then the membranes, these are the membranes, and the placenta, it is the placenta. Okay, it is expelled from the uterus. It is also known as what? Labor. Okay, parturition is the act of childbirth. Okay, now we'll see how this parturition takes place. Okay, when the fetus is fully developed, it pushes down the birth canal. This is the birth canal. Okay, when the fetus is fully developed, it pushes down the birth canal. Okay, when it pushes down the birth canal, then stretching of the cervix takes place. Then the nerve impulse generation takes place in the cervix and it goes to hypothalamus. Okay, generation of nerve impulse from the cervix wall and it goes to hypothalamus. Then hypothalamus stimulate posterior pituitary, right? Posterior pituitary secretes what? Oxytocin. Oxytocin, it acts on uterus, specifically the myometrium layer. Okay. Then the contraction of uterus takes place and it leads to parturition, that is childbirth. Right? It is also known as what? Fetal ejection reflex. Then we will see what is the meaning of lactation. Lactation, it is a process of making human milk. Okay, it is the process of making, not only making and secreting also. secreting milk. Okay. So lactation, it is the biological process through which the female mammals produce and secrete milk from mammary gland to feed their young. Okay. What are the process involved in lactation? First one is mammary gland development. During pregnancy, the hormonal changes stimulate the growth and development of mammary gland in preparation of milk production. Then milk production is also known as lactogenesis. After childbirth, the hormonal changes, particularly the release of prolactin. This is again a potent question. Okay, milk production takes place by prolactin hormone from pituitary gland, which triggers the production of milk in the mammary gland. There are two words. One is milk production by prolactin. Second one is milk ejection by oxytocin. Okay, milk rejection, that is let down reflex. This is not important. Okay, the hormone oxytocin also released from the pituitary gland. Okay, particularly posterior pituitary. Prolactin from anterior pituitary, then oxytocin from posterior pituitary. It causes muscles around the milk producing alveoli to contract. The function of oxytocin is to contract. Like in parturition also, it is contracting the myometrium layer of uterus, and in lactation, it is contracting the alveoli. Okay, so that it pushes the milk through the duct into the nipple. Okay, then the last step is suckling. Okay, when the infant sucks, it stimulates the nerve ending in the nipple. Further, it promotes the release of prolactin and oxytocin, thus continuing the cycle of milk production and ejection. So here you can see it is a mammary gland. It constitutes of adipose tissue, that is the fatty tissue. Then these are the alveolus. Alveolus consists of alveolar cell. Okay, alveolar cell, here the milk production takes place. Okay. Next question is, what is alveolus? It is a modified sweat gland. It is also a PYQ. Okay. What? Alveolus, it is the modified sweat gland. Okay. Then alveolus, it leads to lactiferous duct. Then lactiferous duct to here nipple. Right. Then here we know what? One is parturition process. Second is lactation process. Next up, we have question number 9. That is, what is precocious germination? Precocious germination, it is also known as what? Premature germination. Okay. And it is a process of vivipary. Okay. In plants, vivipary, that is the precocious or premature germination. It is the germination where the seed, while still on parent plant, it gets germinated. You can see these are the seeds. Okay. This is the process of germination. Okay. Germination of seed while it is still on the parent plant. It is known as what? Precocious germination or premature germination. Okay. Why it is taking place? Because it lacks the seed dormancy period. Okay. It skips the seed dormancy period. Okay. What is written here? The immature embryos were removed from the seed and cultured midway through the development. It is not needed. The embryos germinated precociously, that is, without passing through the normal dormancy. When ABA, that is abscisic acid, was added to the medium, then precocious germination didn't occur. That means ABA, which is a plant hormone, it induces what? Dormancy. Right? Then precocious germination, Now we'll see some of the examples of precocious germination are, first one is red mangrove, second one is rhizophora. They skip seed dormancy. Okay. Next up we have what is entomophily. Okay. Entomophily, as you can see from the picture, it is the insect pollination. That means the pollinating agent is insect. Entomophily means insect pollination. Okay. That is, insect is the pollinating agent. Okay. So what are the characteristic feature of a flower that is pollinated by insects? First feature, they are bright in colors. Okay. Sometimes they reward the insects with they reward the insect with pollen and nectar, so that the insect will be coming back to them. Okay. Next, entomophily, it is a insect pollination. Next up we have question number 11, that is what is blood, plasma, human circulatory system and double circulation. Okay. As you know, what is blood? It is the circulating fluid. It is the circulating fluid. Okay. Blood. What are the parts? One is plasma. Second is formed element. Plasma, it consists 55% of the blood. Okay. It is a liquid component. Then formed elements, it comprises 45%. Okay. What are the formed elements that are present in blood? First one is RBC, the cells, WBC, and the platelets. These are coming under formed elements. Next is plasma. Plasma consists of, here you can see, first one is salt and minerals, then water. Water contain 92%. Okay. Then proteins, the vital proteins, they are 7%. What are the vital proteins that are present in plasma? First one is albumin. Second one is globulin and all anti-hemophilic factor. Okay. Then the mineral contain 1%. Okay. Apart from that, they also contain sugar, fats, hormones. These are negligible in quantity. Okay. Also vitamins. Okay. So blood, plasma 55%, formed element 45%. They also formed elements in which RBC, WBC and platelets, they are the formed elements. Then plasma contain salt and minerals 1%, water, which is the most 92%, then proteins, albumin, globulin and all anti-hemophilic factor. Okay. This consists 7% of plasma. Okay. Now this is the test tube. This is a centrifuged blood. That means the RBC are settled down. RBC, that is 45%. This is the 1% that is buffy coat, which contains WBC and platelets. Together, buffy coat and RBC, they are what? They are formed element. Okay. Then 55% that is plasma. Right. Next up we have double circulation. Okay. As you can see from the word double, that means what? That means the blood passes through the heart twice. Okay. That's why it is double circulation. That means the blood passes through the heart twice. Okay. Why double circulation is important? Because it allows the oxygenated blood to be separated from deoxygenated blood. Okay. Here you can see all the deoxygenated blood from the tissue that are carried to right atrium through vena cava. Okay. From the right atrium, it goes to right ventricle through tricuspid valve. Right. Then from the right ventricle, the blood passes to the lungs through pulmonary artery. Okay. Pulmonary artery, it is the only artery that carries deoxygenated blood. Okay. In lungs, then oxygenation of blood takes place. Okay. Then the oxygenated blood, it comes to left atrium via what? Pulmonary vein. It is the only vein which carries what? Oxygenated blood. Right. Then from the left atrium, the blood comes to left ventricle through mitral valve, or also known as bicuspid valve. Okay. Then the left ventricle which pumps out the blood through aorta. Okay. It is the mechanism of double circulation. Here you can see two circuit. One is pulmonary circuit. Second one is systemic circuit. Okay. The blood passes through the heart how many times? Twice. Right. Then one is human circulatory system. Human circulatory system, these are consist of what? Blood vessels that carries blood away from the heart and towards the heart. Which vessels that carries blood away from the heart? That is arteries. Even the pulmonary artery, it carries blood away from the heart to lungs. Right? The arteries then carry blood away from the heart and the vein carry blood towards the heart. Okay. Next question we have question number 12. What is eubacteria? You know the kingdom Monera, right? Monera, these are first point, they are unicellular. Second point, these are the only prokaryotic kingdom. They are only prokaryotic unicellular kingdom, right? Karyotic unicellular kingdom, which is Monera. Okay. Monera, that is only consist of bacteria. Then bacteria also, they are divided into two groups. One is Archaebacteria. Second one is eubacteria. Okay. Archaebacteria, then eubacteria. Okay. Here we will see about eubacteria. First, they are unicellular and prokaryotic microscopic cell. Okay. Then here also you can see first one is prokaryotic cell. That means they lack true nucleus and membrane bound organelle. This is the feature of prokaryotic cell. Okay. Second point, that is cell wall composition. Cell wall composed of what? Peptidoglycan, which provides structural support and protection. Okay. Then bacteria, they have diverse shape. Here you can see these are the diverse shape of bacteria. That is Bacillus, Spirillum or Spirilla, then Cocci. Okay. These are the round shaped, elongated shaped. Okay. So bacteria, they have diverse shape, including what? Cocci, these are the spherical shape. Bacilli, these are the rod shape. And spirilla, they are the spiral shape. Okay. Then we'll know about their reproduction. They reproduce asexually, that is through binary fission. What is binary fission? The parent cell, it will divide it into two identical cells. Right? Then the fifth point is metabolic diversity. Okay. They can be autotrophic. That means they will prepare their own food through photosynthesis or chemosynthesis. Or they can be heterotrophic also. That means they will obtain their food from external source. Okay. Then sixth point is their habitat. Eubacteria, they are found in nearly every habitat. Okay. But archaebacteria, they will be found in harsh habitat. Found in, okay, eubacteria, they found almost everywhere in earth, including what? Soil, water, inside the bodies of other organisms, right? Then seventh one is what are the roles they play in the ecosystem. First, they play crucial roles such as decomposition, then nitrogen fixation, and as symbionts. Okay. You can see the structure of eubacteria. These are the pilus or pili. Okay, this is the plural form. Or flagellum. This is the singular form. Flagella is the plural form. They are the locomotory organs. Okay. Next, eubacteria, it contains a capsule. Then inside the capsule, it has a cell wall. Then inside the cell wall, it has a plasma membrane. It is the genetic material, that is nucleoid. It contains ribosome. Which ribosome? 70S ribosome, because it is a prokaryote. Then it also contains plasmid. Okay. It is the extra genetic material. Okay. Extra genetic material. Okay. It is all about eubacteria. Next question we have, what is the basis of classification of algae? Okay. Algae, what are the basis? Here you can see it is the summary that I have taken from NCERT. You can see what are the classification, basis of classification. First one is their photosynthetic pigments. Okay. Second one is their nature of reserve food. Third one is nature of cell wall components. Fourth one is type, number, and attachment of flagella. Fifth one is their cell structure. Here you can see all the point. First is what are the classes of algae? First one is chlorophyceae, which is the green algae. Second one is phycophyceae, that means the brown algae. And third one is rhodophyceae, that is red algae. What are the major pigments then? In green algae, it contains chlorophyll A and B. Okay. You can see A is common in both all the algae. Okay. Chlorophyll A is common. Then green algae contains chlorophyll A and B. Brown algae contains chlorophyll A and C. Then fucoxanthin. Then red algae, it contains chlorophyll A and D and phycoerythrin. Okay. What are the stored food then? First, starch is the stored food in chlorophyceae. Mannitol and laminarin, they are stored food in phycophyceae. And the Floridian starch, it is the stored food in rhodophyceae. Okay. Cell wall, it composed of cellulose. Here cellulose and algin. In rhodophyceae, it contains cellulose, pectin, and polysulfate ester. Then we'll see the flagellal number. Flagella, number and their position. In chlorophyce, 2 to 8 number of flagella, they are equal and apical. In phycophyce, it is 2 in number, but it is unequal and lateral. Okay. In rhodophyce, no flagella are present. Okay. You can see the habitat also. Okay. What are the star mark question here? Here, you can expect question from measured pigments and stored food. Okay. Okay, so this much for today. I will conclude the session here. Okay. Thank you, everyone.